Hi, my talk today is on the metabolomic and exposomic analysis of autism spectrum disorder, the ripple effects of our changing chemosphere. And I wanted to thank the Brain Foundation for supporting some of this research. So here's a summary slide of the research over the last 10 years, from 2010 to 2020, that to understand autism, we need to understand the cell danger response. My disclosures are here. You can take a look at them on the recorded PowerPoint. And then the public resources and references include a collection of 26 scientific papers that have been published over the past 50 years on the relationship of autism to purinergic signaling, cell danger response, and healing and recovery. And those you can get at this link to our website, item number 11, there's a PDF you can download. And also you can go directly to the journal Mitochondria and look at their most downloaded articles. And 4 of the top 10 are papers on mitochondria, autism, the cell danger response, and healing. So, big question number 1, are the behavioral symptoms of autism caused by a treatable metabolic syndrome? Big question number 2, can this metabolic syndrome be triggered or maintained by environmental chemicals, our exposome, that we can measure in the blood? What do we know from the animal models so far? Well, it turns out, interestingly, whether you choose a genetic model like Fragile X or a number of different environmental models like the maternal immune activation model that can be produced by double-strand RNA or ATP injection or valproate in mouse and rat models, that in the model, there are ASD-like behaviors, synaptic abnormalities, cerebellar abnormalities, metabolic abnormalities, proteome, cytokine, and microbiome abnormalities. But a single kind of treatment, anti-purinergic therapy, actually has been shown to completely correct those abnormalities, whether they're genetic or environmental. So they're normalized behaviors, synapses, cerebellum, metabolism, proteome, cytokines, and microbiome after purinergic signaling therapy with drugs like Suramin, for example. So that leads naturally to a big question number 3. Could ATP signaling or purinergic signaling be the final common pathway that triggers the cell danger response after any kind of environmental or genetic stress? So I emphasize this as, environment and genetics working together. So let's move to the next section of the talk, and this is on mitochondrial functional changes that are needed to fight infection, learn, and to heal. So in the beginning, we start with mitochondria in cells. These are cells down here in the lower left. And there's a network of connected mitochondria, stained red here, that is present in cells that are healthy. As soon as there is any kind of injury, the mitochondrial network fragments and there's a transformation to a different kind of mitochondria that is absolutely necessary to initiate the first stages of healing. But once those stages of healing are completed, the network is reconnected and health is restored. But what happens if there's a block to the healing process? Well, that can lead to a repeating cycle of cellular defense that can lead to chronic illness in 60% of adults, and now in the US 40% of children live with some kind of chronic illness. So why are purines so important? And purines, the asterisk here means ATP, ADP, AMP, xanthine. These are molecules that are used by the cell to make RNA and DNA, and also for energy metabolism. So if you look at all the 1,200 proteins in mitochondria, 70% of them are enzymes that catalyze biochemical reactions. And then over half of those enzymes turn out to be regulated by purines. So extracellular ATP is a key regulator of the cell danger response. And another interesting thing that's become, that's become known over the last 10 years has been that all stress cells leak ATP. So the more stress a cell is under, the more the channels in the cell membrane that will conduct nucleotides and other metabolites out of the cell, for example, this pannexin P2X7, will open up and more ATP and metabolites are released, where in the extracellular space, they act as metabokines, signaling molecules that can bind to receptors on the cell surface. So mitochondria are constantly surveilling the chemistry of the cell, both internally and through external circuits, and talking to the nucleus, what we call the short path retrograde signal. The nucleus talks to mitochondria, and if those channels begin to open up and we lose too much ATP, then the available ATP within the cell is reduced, and the functional capacity of the cells decrease, its ability to heal is decreased. And then outside the cell, ATP acts as an inflammatory signaling molecule. So we went looking to try to find, is there a way to actually block the loss of that ATP? And it turns out there is, but that's a talk for another day. So what about, if the cell danger response is so universal, can we see it in plants? And the answer is yes. And so it involves, as it does in humans, the release of intracellular calcium stores. And in this case, here's a little mustard plant that has a little bite taken by a cricket, simulated by in the laboratory. And it turns out if you add either glutamate or ATP to that little bite, that injury, the signal of that injury is transferred to the rest of the cell within 2 minutes. Okay, so if we try to make sense of this, put it into context, what we find is that three different kinds of mitochondrial structures and function are required to heal from any injury, and we call these mitochondrial forms the M1, M0, and M2 forms of mitochondria. So this is the normal healing cycle. And so typically we start a day, we wake up, we have equal activity, we have nutrient intake, it's a little bit of stress, so there's a little bit of extracellular ATP released, but that actually is necessary to help facilitate restorative sleep and adaptation. And we go through this cycle. If the injury is more severe and the stress is more severe through toxins or infections or adjuvants, metals or pollution, then what happens is that mitochondria change their structure. So here's an example of the normal filamentous mitochondria that I've been exposed to a stress in cell culture, and a video is being taken of them undergoing what we call the spaghetti to meatball transformation, or this transformation from M2 filamentous form to the M1 fragmented form of mitochondria. Those fragmented forms of mitochondria are called the M1. They undergo glycolysis. After the injuries have been contained and the infection has been contained, then cells have to be replaced in a proliferative phase that requires M0 mitochondria. And ultimately what we find is there are checkpoints that identify the transitions between these three different types of mitochondria, M1, M0, and M2, that ultimately can be traced back to a number of different cytokines and metabokines, and we're going to be focusing on extracellular ATP. So initially you start with increased extracellular ATP, and you complete the healing with decreased extracellular ATP. So now we're going to move on to metabolomics and exposomics. So we think of drawing a sample of blood like taking a drop of water from an ocean or a river ecosystem, and we can look at the natural molecules, and we can look at the man-made molecules in the exposome. And this is how we do it at UCSD. We have nearly 700 natural molecules that we target from over 63 different biochemical pathways. And then we also have an exposome panel that looks at over 1,000 different man-made molecules, including pesticides, flame retardants, phthalates, and plasticizers, personal care products, and even mycotoxins and variety of bacterial toxins. Overall, there's about 1,900 chemicals that we measure. So now I'm going to tell you about a study that we did in collaboration with Judy Vandewater at the UC Davis Mind Institute that looked at children, the rough 4 to 6 year old children that were getting their routine pre-kindergarten vaccinations. Okay, so we drew blood at baseline from 22 typically developing children and 31 children with ASD. Those children received routine vaccination. Blood was drawn 2 days afterwards, and daily temperatures were taken for a month. Now today I'm only going to talk to you about the differences we see at baseline between typically developing children and ASD. And then we'll also talk about daily temperatures. And I'll talk about the response to immunization in another talk later on. Okay, so children with ASD have an altered, what we call the triphasic temperature response to vaccination. So interestingly, so the red line here are children with ASD, and the blue line are children that are typically developing. And we found at baseline that the children with autism had a 0.7 degree Fahrenheit lower body temperature. After routine immunization, both groups increased their temperature. It's not a full fever, but they both increased. We call that the first phase. The second phase is a rebound reduction in temperature or hypothermic response. And then the third phase is this rebound hyperthermia or normothermic response in the typically developing. So the blue, basically they go up, down, and recover. And they're the same, from about day 5 on, they're back to their baseline temperature. But the children with autism go up, down, up, and then they have a rebound hyperthermia where for a whole month after a single vaccination, they have a temperature that's about 1.2 degrees Fahrenheit above the temperature before they received immunization. Okay. All right. So what happens if we look at the baseline differences in the metabolome of children with autism and typically developing children? So here are the children with ASD. Here are the children with that are typically developing controls. These are again 4 to 6 year olds, males and females. And when we use metabolomics, just looking at 4 different things, we can have a diagnostic accuracy for the identification of children with ASD of about 88%. Okay, with about a 90% accuracy. So what are some of the chemicals? Well, this is a list of some of the different chemicals that are abnormal: the ceramides and phosphatidylcholines, isoleucine, methionine, methylcysteine, adenine. So anyway, overall summary is that the children with autism had decreased lipids of many kinds: ceramides, sphingomyelins, and PC lipids. They had an increase in branched chain and aromatic amino acids and methionine, an increase in methylcysteine. Histamine increased, as you might see with activation of mast cells. Increased citrulline and adenine, and an increased endocannabinoids, and a decrease in the in ADP. Okay, so it turns out we know from previous studies that these are each markers of a purine-driven persistent cell danger response in children, and some of this has been actually reproduced independently in children, not just mice, in a study by the Persico group in Italy. So if you look at Fragile X syndrome, you also see that the top pathway that carries the most metabolic variants is purines. Okay, so now I'm going to shift to the exposome. Okay, so we think of these as kind of footprints of the interaction of the chemosphere with our metabolism. And so we ask the question, what happens when you measure 1,000 man-made chemicals in the blood of these children that are age 4 to 6 years of age? So what we found was the average child had 39 different man-made chemicals in their blood. It turned out there was no actual difference in the number of chemicals in ASD and TD groups. And overall, there were 27 chemicals out of the 1,000 that we measured that were present in 75 to 100% of the samples. And to analyze this, exposomics requires a little bit different statistical analysis. So there are two different kinds. So for the chemicals that are common to all the samples, you can actually look at the magnitude of those chemicals in the blood by common measures of students' t-test and Mann-Whitney or Welch's t-test. But to look at the frequency of chemicals, the counts in children with autism versus the counts of those chemicals in typically developing kids, you actually have to use another test called Fisher's exact test or a chi-square. So the other interesting thing is that the Environmental Protection Agency establishes what's called a NOEL level for many, well, I won't say many, some environmental chemicals. And NOEL stands for No Observable Adverse Event Level. Okay, so it turns out this is, these are not, these are present for some of the chemicals that we know a fair amount of, for example, some of the Teflons, phthalates, but for a lot of the other chemicals, we don't have good NOEL levels yet. And in general, our mass spec methods allow us to measure between 100 to 10,000 times below published NOEL levels. So typically what we find in the children is the presence of chemicals that are far below what would normally be considered to have a toxic effect based on studies in rats and mice. So if we just focus on what we found on the 27 chemosphere chemicals that were present in virtually all the samples, this is what we find, is that the perfluorononoic acid, or PFNA, which is a kind of Teflon, was actually decreased in children with autism compared to controls. And dibenzacridine, which is an exhaust product and also a carcinogen, was also decreased in children with autism. And then a common food additive or preservative called terse-butyl hydroquinone actually was present in 19 of 31 children with ASD, but 20 of 22 children with TD. So it was actually decreased in children with autism, and it was also statistically significant. But if you look at maybe the bigger message of this is that there are dozens to hundreds to potentially thousands of chemicals that are detectable in the blood if you look. We looked for 1,000, but there are many thousands more that could be looked at. And what we find is this is the tip of the iceberg. So in the blood, we can find pyrethroid insecticides, food preservatives, plastic wrap, hydraulic fluid chemicals, fungicides, dyes, plasticizers, many different Teflons, herbicides, endocrine disruptors, some of the phthalates. So these are ubiquitous molecules in the environment today. And it's just the tip of the iceberg because we didn't look for everything that we could have. So here's an example that PFNA, the perfluorononoic acid, was lower in ASD. This is what it looks like over on the right-hand side. This is the structure of this Teflon. You can see over on the left, it was lower than in TDs. And PFNA turns out to be the most common Teflon found in Atlantic dolphins and polar bears. And the levels can be 400 parts per billion in polar bears. But in New Jersey, the new standard in municipal water is it has to be below 13 parts per trillion, okay? So it means that the blood levels that were in the polar bears and dolphins can be 10,000 times above what was considered safe in the drinking water. And the dibenzacridine was also lower in ASD. You can see lower than in ASD than TD. This is what it looks like. It's a planar molecule that is a polyaromatic hydrocarbon. And this is the terse butyl hydroquinone result. So this is what it looks like on the upper right of your screen. It's also less common. It was present in 61% of children with ASD, but 91% of children with that were typically developing controls. So where does this leave us? Well, it leads us to this question: could children with ASD be more sensitive to the same chemicals compared to TD children? So what's the evidence for that? Well, there were no chemicals in our list of over 1,000 that were statistically increased in ASD. 18 of 27, or 70% of them, trended lower. 7 of 27 were unchanged. And just the two that were statistically significant by magnitude were actually decreased, not increased. So how does this fit in with the published literature? So it turns out that this has been seen in other studies. So other studies have found decreased toxins and heavy metals in children with autism compared to controls. For example, a nice paper by Jim Adams that looked at heavy metal and trace metal levels in hair samples from children with autism. They were actually lower than in children that were typically developing controls. And so, although one explanation might be that's failed excretion, an alternative explanation is that by activation of the cell danger response, there was successful sequestration of those dangerous metals. And it turns out in the paper that's just published this year, there's also, when they looked at polychlorinated biphenyls, the PCBs, in the blood of children in Jamaica, there's also a negative correlation. In other words, the higher concentration of PCB made it less likely the child had ASD, so with an odds ratio of 0.44. Well, so how does this fit again with the cell danger response? It turns out, when we describe the metabolic features of the cell danger response in a paper that came out in 2014, when we were looking at metals, we noticed that in the danger side of the response, there was actually increased sequestration as a biological response to toxic threat, to sequester these potentially toxic metals away from sensitive organs like the brain. Okay, so this leads naturally to a corollary question. Could the restricted food preferences, choices seen in over 80% of children with ASD be driven by autonomic behaviors that were evolved to decrease the exposure to foodborne chemicals that would otherwise trigger the CDR, and an anxiety response from increased purines and ATP and xanthine? So what if those environmental changes found associated with autism are not specific to autism, but instead act as an invisible rising tide that increase the risk of dozens of other childhood disorders and adult chronic disorders? So here's a model. If you look at the total chemical load, in air, water, food, and medication. So Martha Herbert is one that really drew attention to this over 10 years ago about this environmental toxic load. But some of the chemicals and the levels of chemicals don't have to be demonstrably toxic. They can just be sensed and reacted to by children who have particularly sensitive mitochondria. So after fertilization, if you imagine all these all these dots being children, and that some children are born with the blue dots that have increased ability of their mitochondria to sense and respond to the world. If you then place that children in a high chemical load environment, then the most sensitive children will detect and respond, activating the cell danger response to change mitochondrial function in order to facilitate healing and resistance initially to that threat. So potentially what happens is that you take all those more sensitive children. Some of them will respond ultimately through a combination of genes and environment and lead to autism. But other combinations can lead to asthma and diabetes and cancer and OCD, inflammatory bowel disease, and gluten sensitivity. So that this persistent activation of the cell danger response or blocks in the healing cycle might help to explain why 40% of children now live with chronic disorders. But 35 years ago, for example, when I was in medical school, this number was 5% of children were living with these very same disorders. And it's not because physicians back in 1985 were unable to diagnose autism, asthma, diabetes, or cancer, or any of these other things, okay? It was because they were less prevalent. Okay, so this leads us to some closing questions. What if not all children are equally sensitive to the environment? These concepts of sensitivity and resilience are familiar, but have not been applied quantitatively in environmental toxicology. So what if the environmental stresses triggered, the very same amount of ATP, but children with ASD were more sensitive to this ATP or purinergic signaling? What if we knew a way in which we could identify children who are more sensitive? Could we then intervene in the first few months of life to make them more resilient and prevent lifelong symptoms and disability? Well, this is how I'll close my talk today, and this is a plea to everybody watching this, that if you would like to help us discover how sensitive children might be identified at birth, and if you have a child that's 3 to 10 years old who's born in California, either with ASD or typically developing child, go to this website, go to this link, naviolab.ucsd.edu/study. There are no new blood samples required. All this is we getting parent permission to go back in time to the dried blood spots that were collected and stored at the time of birth of your child in California, to measure over 1,000 different chemicals in those dried blood spots. So there are no new samples required. You get a $40 Amazon gift card, and it takes only 15 minutes to fill out the forms. So I'll conclude there, and with a page that gives thanks to our financial supporters. It starts with the UCSD Christini Fund that has made everything that we've done over the past 20 years possible. But also includes the Brain Foundation and CHARM. And I thank all of our supporters and you for your attention. Thank you.